Serum immunoglobulin and the significance in blood banking. The immunoglobulin or glycoprotein molecules that are produced by plasma cells in response to the immunoglobulin, and these functions as antibodies. Basic function of immunoglobulin: heavy and light chains. The basic immunoglobulin unit consists of two identical polypeptide heavy chains with heavy molecular weight 55,000 to 75,000 molecular weight, and two identical polypeptide light chains with low molecular weight 25,000 molecular weight. The immunoglobulin are named according to the structure of heavy chains. These five types of heavy chains are designated as alpha IgA, delta IgD, epsilon IgE, gamma IgG, and mu IgM. The light chains are of only two types, kappa and lambda. The two light chains of every immunoglobulin will be identical, either kappa or lambda. Interchain bonding: the two heavy chains are held together by interchain bonding covalent bonds, disulfide bond, and by non-covalent bonds. Within each chain there are also interchain disulfide bond, variable and constant region. Each light chain consists of 220 amino acids, and each heavy chain has 440 amino acids. The first 110 to 120 amino acids of both heavy and light chains have a variable sequences and forms variable regions, which is considered to determine the antibody specificity. The rest of the light chain and heavy chain represents the region with the constant amino acid sequence. Hinge region: this is the region at which the arms of the antibody molecules forms a Y. It is called as a hinge region because there is some flexibility in the molecules at this point. In this region, the enzyme and albumin, the bonds in this region are all covalent bonds. Fragments: each fragments of immunoglobulin is divided into three parts, one FC and two FAB pieces. The FC is insoluble fraction, which is crystallized in cold. C for crystallizable, and FAB is soluble fragment, which is having ability to bind antibody for antigen binding. Production of immunoglobulin: the immunoglobulin makes up approximately 20% of total serum. The introduction of red cells antigen into the circulation of an individual lacking that antigen may stimulate the production of a corresponding antibody. This may occur as a result of blood transfusion therapy or feto-maternal blood group incompatibility in pregnancy. These are called incomplete or acquired IgG, mostly immune antibody or IgG that reacts best at 37 degrees centigrade and requires the use of a anti-human globulin serum for detection. The common immune antibody reacts with the RH, Kell, Duffy, Kidd, and SS blood group system. Certain antibody occurs without known antigenic stimuli. These are known as a complete or natural antibody, IgM, IgA. The commonly naturally occurring antibody reacts with the ABO, HH, LI, Lewis, MN, and P blood group system. Classes of antibody: IgG provides a long-term immunity or protection. IgM, first antibody produced in response to the antigenic stimuli. IgA, it is found in secretions like tears, saliva, colostrum, and mucus. Protects against infection in urinary, gastrointestinal, and respiratory tract. IgE involves in allergic reaction. IgD, not much known about its surface receptors of B lymphocyte. The most important classes of antibody in blood banking are IgM and IgG, and to certain extent IgA. The characteristics of IgG and IgM antibody significantly in blood banking. Immunoglobulin have a great importance in immunohematology. Approximately 1/3 of anti-A and anti-B are IgA, and 2/3 are IgM, and occasionally IgG. IgG can cross placenta and hence play an important role in hemolytic disease of newborn. The IgM antibody shows a visible and agglutination, hence plays an important role in blood grouping. The clinical significance of a red cell antibody in blood bank depends on whether they can cause in vivo hemolysis, which in turn will cause a transfusion reactions or hemolytic disease of newborn. The IgG will frequently causes in vivo hemolysis due to the antibody coating the red blood cells. IgM, with a few important exceptions, usually does not cause in vivo hemolysis. The most important of these exceptions are ABO antibody. The size of antibody: IgG and IgE are relatively small since it comprises of only one immunoglobulin subunit monomer. The IgG is Y-shaped and IgA is T-shaped. IgM is relatively large since it is comprised of 5 immunoglobulin subunit pentamer. serum concentration. IgG is found in the largest concentration of all immunoglobulin in the plasma. IgM is found in relatively small amount. IgA is the second most common serum immunoglobulin. IgG is greater than IgA is greater than IgM. Complement fixation. IgG will do if condition are optimal. IgM very good complement activator. IgA does not fix complement unless aggregated. Placental transfer. IgG is small enough to easily cross placenta, and it is the only immunoglobulin capable of doing so. IgM, IgG and other classes do not cross placenta. Optimum temperature of reactivity. IgG is 37 degrees centigrade, IgA 37 degrees centigrade, IgM 4 degrees centigrade. May react at any temperature below 38 degrees centigrade. Number of antigen binding site. IgG has 2 binding site, IgM has 10 binding site. General function of immunoglobulin. The antigen binding. Immunoglobulin binds specifically to one or few closely related antigen. Each immunoglobulin actually binds to a specific antigenic determinant. The antigen binding by antibody is a primary functions of antibody and can result in protection of host. The valency of antibody refers to the number of antigenic determinant that an individual antibody molecules can bind. The valency of all antibody is at least 2 and in some instant more. The fixation of complement. This results in lysis of cell and releases of biologically active molecules. Binding of various cell types. The phagocytic cells, lymphocytes, platelets and basophils have receptors that binds immunoglobulin. This binding can activate the cells to perform some functions. Some immunoglobulin also binds to receptor on placental trophoblast, which results in transfer of immunoglobulin across the placenta. As a result, the transferred maternal antibody provides immunity to the fetus and newborn. Clinical significance of immunoglobulin. IgG. Increase in chronic granulomatous infection, hyperimmunization, liver disease, malnutrition, severe dysprotein anemia, disease associated with hypersensitivity, granulomas, dermatological disorder, and IgG myeloma, rheumatoid arthritis. IgG is decrease in lymphoid aplasia, selective IgA deficiency, IgA myeloma, Binchon's protein anemia, chronic lymphoblastic leukemia. IgM is increased in adult in trypanosomiasis, actinomycosis, malaria, infectious mononucleosis, lupus erythematosus, rheumatoid arthritis. In newborn, a level of IgM above 20 nanogram per deciliter is an indication of utero stimulation of immune system and stimulation by rubella virus, cytomegalovirus, syphilis or toxoplasmosis. Decrease in A gamma globin anemia, lymphoproliferative disorder, certain cases, lymphoid aplasia, IgG and IgA myeloma, chronic lymphoblastic leukemia. IgA is increased in certain stages of collagen and other autoimmune disorders such as rheumatoid arthritis and lupus erythematosus. Chronic infections not based on immunological deficiency. IgA myeloma. Decrease in immunologic deficiencies. State example: dis gamma globin anemia, congenital and acquired A gamma globin anemia, and hypo gamma globin anemia. Malabsorption syndrome, lymphoid aplasia, IgG myeloma, acute lymphoblastic leukemia, chronic lymphoblastic leukemia. Chronic acute lymphoblastic leukemia, chronic lymphoblastic leukemia. IgD is increasing in chronic infection, IgD myeloma. IgE is increased in atopic skin disease such as eczema, hay fever, asthma, anaphylactic shock, IgE myeloma. IgE is decreased in congenital A gamma globin anemia, hypo gamma globin anemia due to the faulty metabolism or synthesis of immunoglobulin.